Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the third unit of Plant Biotechnology course. In this unit, we will discuss about the protoplast preparation and their applications like somatic hybridization, and then we will also learn about the somaclonal variations and their applications. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. This project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. In this lecture, I will talk about the protoplasts, various methods of protoplast isolation, requirements for in vitro culturing of the protoplasts, and finally, there are various applications of the protoplast in basic biological sciences and their commercial applications. So, protoplasts. A plant cell, when stripped of its cell wall, is called as a protoplast. These are also called as naked plant cells, referring to as all the components of the plant cell except the cell walls. Here is a cartoon of the plant cell where cell wall and plasma membrane are clearly annotated, and there are several other organelles like vacuoles, ribosomes, nuclei, endoplasmic reticulum, Golgi, and so on. When this cell wall is removed, everything else other than the cell wall is called as a protoplast. Though protoplasts may be prepared from plant cells of diverse shapes like rectangular or polygonal types of cells, however, the protoplasts are usually spherical in shape. So the shape of the plant cell is because of the rigidity of the plant cell. Sometimes these are also called as the functional plant cells without the cell walls, and they are supposed to be performing almost all the functions of the plant cells. However, absence of the cell walls may affect the metabolism, and it may introduce ultrastructural changes in the protoplast. It may affect the permeability of the cell membrane that may result in the leakage of solute from the protoplast, and hence it may compromise or affect the physiological status of these protoplasts. Protoplasts are very fragile both biochemically as well as mechanically. In the plant cells, it is protected by the presence of the cell wall. Once it's removed, it is the membrane that has to hold everything. Hence, these are fragile and can easily be broken. Therefore, special care is required for handling the protoplast. Further, the fragility of the protoplast may vary with the physiological conditions of the source tissue. fragility can be high if the protoplasts are prepared from the mature or aging tissues. Now, the protoplasts, like the plant cells, are totipotent, and hence they can regenerate into the complete plants. And finally, protoplasts have several applications in basic biological sciences as well as in commercial industry too. We will discuss the application in more details towards the end of this lecture. So first we will discuss what the isolation methods. So in order to prepare the protoplast, we need to cut open the cell wall or need to dissolve the cell wall completely. And depending upon the method used to strip off the cell wall to extract the protoplast from these cells, these can be grouped into two categories. One is the mechanical extraction and second is the enzymatic extraction. So the first is the mechanical extraction of the protoplast. However, before going ahead, it is important to learn about the maintenance of osmotic pressure and plasmolysis. Once the cell wall is removed, how is the protoplast going to behave in the liquid culture? And this behavior of the protoplast would depend upon the strength of the media. Like here are the two solutions. One is the hypotonic solution and one is the hypertonic solution. So theoretically, if the concentration of the solutes or the dissolved content in the solution is less than that of the cytoplasmic content, then it is called as the hypotonic solution. However, if the concentration of solutes or dissolved content is higher than that of the cytoplasmic content or protoplasmic content, it's called as hypotonic solution. Now, what will happen if we place the protoplast into the hypotonic solution? So the protoplast so in the hypotonic solution, the strength of the solution is lower as compared to the protoplast, and therefore they will take up the water, they will swell, and the turbo pressure would increase, finally leading to the bursting of the protoplast. But in the hypertonic, the reverse will happen. Now the water will move out of the cells or the protoplasts to the surrounding media, and the protoplast will shrink. This phenomenon of shrinking of protoplast in the hypertonic solution is called as the protoplast plasmolysis. Therefore, it's very important to maintain the osmotic pressure at the suitable levels to maintain the protoplast in the liquid nutrient media. Usually, protoplast can be maintained easily in a slightly plasmolysed state. Mechanical extraction of the protoplast is a crude procedure. It was first reported by Klerker in 1892. The suitable tissue is selected from which we need to prepare the protoplast. Like here is the tissue from where we need to prepare the protoplast. In the mechanical extraction, we need to cut the tissue with a sharp blade to remove the protoplast. Now here in this tissue, the cells are fully turgid, and it would not be possible to make a cut in the cell walls without damaging the protoplast part of the cell. Therefore, the first step is to plasmolyze this tissue. So for plasmolysis, this tissue is incubated in the hypertonic solution. That is, the concentration of the solute in the solution should be higher as compared to the inside the cytoplasm. Usually it is performed by incubating the tissue in the 1 molar sucrose solution. This results in the shrinking of the protoplasts. So the protoplasts move away from the cell walls, and this will make enough space for making the cuts in the cell walls without damaging the protoplast. Now, plasmolysed tissue is cut or dissected with the sharp blade into the smaller tissue segments, and in the process it will release the protoplast from the cells. Like here in the first case, the cut is made through the protoplast in the first cell, and therefore this protoplast is damaged or it will burst into the solution. However, in the second cell, the cut is away from the plasmolysed protoplast. Hence, this will release this plasmolysed protoplast in the solution when the cut will be made or cell wall will be removed. Similarly, from many cells, intact protoplast can be released in the solution when the several cuts are made to this plasmolysed tissue. However, most of the protoplasts are damaged during this cutting procedures. Practically, mechanical extraction of the protoplast is not of much use, as the efficiency of the mechanical extraction method is very, very low. It results in very low yields in terms of number of protoplasts prepared through the cutting the plasmolysed tissue. Therefore, it is a laborious and tedious method. Even to extract the minimum number of protoplasts, large amount of tissue would be required and need to be processed through the plasmolysis and cuttings. Further, the cells needs to be plasmolysed, and the maximum osmotic shrinking would be required for extracting intact protoplast. This osmotic shrinking can impact the physiological status of the protoplast. Also, it has been found that the viability of the protoplast prepared through the mechanical extraction or mechanical extraction method is usually low as compared to the other methods. So the osmotic shrinking may interfere with the physiological capabilities of the protoplasts. Further, this method of mechanical extraction is restricted to the tissues having vacuolated cells, as the success rate with the other tissues or cells is very, very low. Next is the enzymatic extraction of protoplasts. Enzymatic extraction method is the most popular and widely used for preparation of protoplasts from diverse type of plant tissues. Plant cell walls are the extracellular matrix that surround the plant cells and play several important functions, like these are the building blocks of the stable plant structure and provide mechanical strength to the plants. These are made up of cellulose, matrix polysaccharides, and lignin. However, lignin is usually present in the secondary cell walls, and the protoplast preparations, we use only primary tissues where the secondary cell wall thickenings are absent, and so lignin will not be there. Here is the cartoon structure of the arrangement of the plant cells. If we zoom in at the junction of the cells, we find another layer, that is the middle lamella. So the individual plant cells are joined together by special deposition of polysaccharides. It is called as middle lamella. Middle lamella is a wall that is made up of the pectins and holds or cements the cell walls of the adjoining plant cells together. The major component of the middle lamella are the calcium and magnesium pectate, and therefore enzyme would be required to digest the cell wall components as well as the middle lamella in order to release the protoplast into the media. Enzymatic digestion of the plant cell walls was demonstrated by Cocking in 1960 for the first time, which opened up totally a new chapter in the history of plant science research. Cocking demonstrated that the cell wall digesting enzymes can remove the cell walls and protoplasts can be released into the media. Later, several different types of enzymes from different sources of origin in various concentrations were tested and methods was optimized. That resulted in the significant increase in the efficiency of protoplast preparation using enzymatic digestions. Usually, combination of cellulases and pectinases are required for digesting the cell walls. The enzymes that are required for the protoplast preparation are commercially available. Usually, pectinases and cellulases are the ones which are most commonly used for protoplast preparations. Cellulases are usually used up to 2% of the concentration, and pectinases can be used up to 1% of their concentration for the digestion of the cell walls effectively in most of the plant species. However, in some plant species, it may not be enough, and addition of another type of enzymes, that is the hemicelluloses, may be necessary. So basically, the requirement of the enzyme and their concentration may vary depending upon the cell wall composition that varies significantly among different group of plants. These cell wall digesting enzymes are commercially available as different brand names, and depending upon the source, type of the enzyme and the mixtures, these may have their specific pH and temperature requirements. Usually, most of these enzymes work in the pH range of 4.5 to 6 and the temperature range of about 25 to 30 degrees Celsius. Even plant-specific cell wall digesting enzyme cocktails are also available for few plants. These cocktails have been optimized for the best result in these plant species. Now, based upon how the pectinases and cellulases are used for digesting the cell walls, this method can be grouped into two categories. One is the sequential method, or also called as two-step method, and second is the direct method or a one-step method. So the first is the sequential method or two-step method. So in the sequential method, pectinases and cellulases are used separately, and hence it is also called as two-step method. First, the small segments of the target tissue are prepared. Now, in order to release the protoplast from the plant cells, we need to remove the plant cell walls as well as the middle lamella. Middle lamella is the deposition of calcium and magnesium pactates, and if we dissolve the middle lamella, the individual cells will be released, or the cell walls will be more accessible for the cellulase enzymes to chew. Therefore, the first step is the tissue segments are incubated with the pectinases to dissolve the middle lamella. So, dissolution or dissolving of the or chewing of the middle lamella will release the individual cells. After the digestion of the middle lamella, solution is usually filtered to remove the debris, and cells may be washed to clean. In the second step, the tissues, tentatively the individual cells, is incubated with the second set of enzymes, that is cellulases. In this step, cellulases will digest the actual cell walls and would release the protoplast into the media. Again, the solution is filtered to remove the debris and washed. So in this way, pectinases and cellulases are used one after another in a sequential manner, and hence the method is called as sequential digestion method or two-step method. Next is the direct method or one-step method. So in the direct method, both the enzymes or both group of enzymes, that is the pectinases as well as the cellulases, are used together to digest the cell walls and middle lamella. So the small tissue segments of the target tissue are prepared and are incubated with the pectinases and cellulases in optimal conditions of the pH and temperatures. The time taken to complete the digestion may vary among different plants and need to be optimized. After the digestions are complete, the solution is filtered and the protoplast may be washed to clean up. So next is the purification of the protoplast. Now, after the tissue is digested with the required enzymes, that is the pectinases or cellulases, in addition to the protoplast, the solution mixture may contain lots of debris like undigested leftover materials or undigested cells, digested segments or remains of the broken protoplasts, and enzymes and other components used for the digestion reaction. Usually, the large debris like the cell clumps or the undigested large tissue fragments or tissues can be easily removed by filtration through the nylon mesh of the pore size of about 45 to 55 micrometer. In order to remove the smaller debris or dissolved contaminants like the enzymes and other components released from bursting of the protoplast, mild centrifugation is performed, usually around 500 rpm or even less, where the protoplast will settle down in the palate. Now the supernatant is discarded and it is replaced with the fresh media. These protoplasts may further be washed, that is the centrifugation and resuspension in the washing media may be repeated few times. Further, if the protoplasts are suspended in the osmoticum or hypertonic sucrose solution like sucrose solution of about 20% concentration, after centrifugation, the viable protoplast will float on the top surface of the sucrose solution, forming a band, and these can be slowly or carefully extracted and resuspended in the osmoticum or in a fresh tube using the liquid media of suitable osmoticum. And further they may be washed several times, again resuspending into the washing media before they can be transferred to the culture medium. Now the protoplast solution may contain subprotoplast as a contaminants too. Now what are these subprotoplasts? Now the protoplast may split into two or more smaller entities, and these smaller entities which are formed by splitting of the protoplast are called as the subprotoplast. Here is a plant cell. The plasmolysis of this plant cell will result in shrinking of the protoplast. Further plasmolysis may result into the splitting of this protoplast into two or more subunits. Only one of these subunits would carry the nucleus, and these fragmented segments or subunits of the protoplast are called as the subprotoplasts. Now, depending upon the content of the subprotoplast, like what do they contain, whether the nucleus is present or absent, whether the entire cytoplasm is present or just the part of cytoplasm is present, these may be categorized into different categories. The first one are the karyoplasts. So if the subunits formed by splitting of the protoplast have the original nucleus, which is surrounded by some amount of cytoplasm and the plasma membrane, this structure is called as the karyoplast. These are also called as mini protoplasts. So another one are the microprotoplasts. So if these subprotoplasts, they contain only the fraction of the nuclear material and little cytoplasm along with the plasma membrane, these are called as microprotoplasts. Similarly, the cytoplasts are the subunit structures that has almost the entire cytoplasm of the cell but lack the nuclei. And if only the fraction of the cytoplasm and outer membrane are there without the nuclei, it is called as microplasts. For the culturing purposes, only the complete protoplast should be used. The advantages of the enzymatic method. Now, the use of the cell wall digesting enzymes for the preparation of the protoplast have made it possible to make large-scale protoplast preparations. Enzymatic methods are easier to execute and they does not require any specialized equipment, and yields are much higher as compared to the mechanical methods. The percentage of the viable protoplast is also much higher as compared to the mechanical methods. Further, the osmotic shrinking is required at the minimum level, and therefore percentage of the subprotoplast formed during the protoplast preparations are also much less as compared to the mechanical methods where plasmolysis is induced to the maximum levels. Theoretically, protoplasts can be prepared from any of the plant tissues using enzymatic methods and is not restricted to the vacuolated cells. However, the leaf mesophyll cells and the cells from the suspension cultures are the good sources for the preparation of healthy and viable protoplasts. Next is the protoplast viability. Now, once the protoplasts are isolated, in order to use them for further applications, protoplasts should be intact, healthy, and viable. only the viable protoplasts would be able to divide and regenerate. Therefore, it is very important to test the protoplast viability. There are several methods that can be used to check the viability of the isolated protoplasts. First one is the fluorescent diacetate staining or FDA staining. Now, fluorescent diacetate is a non-fluorescent molecule. Further, it is a non-polar and can permeate across the plasma membrane freely. Therefore, when provided in the culture, it enters into the protoplast readily. It is a non-fluorescent molecule, therefore just the entry of FDA in the cells will not give any indication of protoplast viability. However, it is a substrate for the esterase enzyme which will be present in the viable protoplasts. Esterase enzyme digests the FDA and releases a polar part or polar molecule, that is a fluorescein, which fluoresces green under the UV light. Now the fluorescein cannot permeate freely across the plasma membrane and hence accumulate in the protoplast and can be analyzed under the UV light. In the dead and the broken cells, it will be lost or would not accumulate because of lack of the enzymatic activity. Similarly, Evans blue staining can also be used. However, it stains the damaged cells. It cannot enter the viable cells by itself and accumulates only in the protoplasts that have lost their plasma integrity. Therefore, the dead cells stain blue and the intact or viable cells remain unstained. Similarly, phenolphthalein staining can also be used to stain the dead cells. It stains the dead cells red. In other methods, photosynthetic activity or the oxygen uptake of the protoplast can be measured as an indication of the viability of the protoplast. Or simply analyzing the ability of the protoplast to undergo mitotic divisions can be a good indication of protoplast viability. Next is the protoplast culture. In order to culture the protoplast, the first step would be to induce the cell wall formation. During the culture, most of the protoplast isolated from differentiated cells would undergo a rapid dedifferentiation, while at the same time initiation of synthesis of new wall will also start. Several parameters that can affect the protoplast culture includes the physiology of the source tissue or the plants from where tissues were harvested, culture in the liquid media or in the semi-solid state, then the composition of the nutrient media used, and finally the osmoticum status of the culture media. So the first one is the physiology of the source tissue. Earlier in the first unit, we discussed the various biological factors or parameters of the tissue types or the donor plants that can impact the regeneration efficiency in the in vitro conditions. Similarly, these factors impact the responses of the protoplast too in the in vitro cultures, like the genotype of the plants. The regeneration efficiency of the plant is genetically regulated, and the inherent capability to the in vitro regeneration responses may naturally vary among the different plant species. Therefore, preparation and culture of the protoplasts from one species may be easier than the other plant species. So, depending upon the inherent regeneration capability, more optimization may be required if optimized protocol do not work. Similarly, the health of the donor plants of the source tissue and the metabolic status of the plants directly affect the culture and regeneration efficiency of protoplasts. Physiology of the plant development or the metabolic or mitotic status of the source tissue changes with the environmental conditions. During the favorable growth conditions, plants grow actively and explants from these mitotically active tissues would perform much better. But during the unfavorable conditions, when the plant tissues are almost dormant, that is, very little metabolical activity is there, or these will be mitotically inactive, their responses to the in vitro culturing will be compromised. In addition to the inherent capabilities of the source tissue, donor plants or the source tissue may be pre-treated to enhance the efficiency of protoplast preparation or the culturing capacity. These may include, first one is plasmolysis. The treatment of the source tissue for plasmolysis can help in improving the efficiency of protoplast preparation. As we saw earlier in the mechanical extractions, similarly it can be helpful in the enzymatic methods too. However, the abrupt change in the size and shape of the protoplast can impact the subcellular architecture. Further, plasmolyzing conditions usually suppress the photosynthetic capacity and even the respiration rate too, and therefore it can impact the responses of the protoplast in the culture media. Also, the incubation of the source tissue at low temperatures have also been reported to improve the efficiency of the protoplast culture. Similarly, donor plants are sometimes treated with phytohormones to induce the metabolic activities and hence impact the physiological status of the tissues. These may enhance the chances of the recovery of viable protoplasts as well as their culture efficiencies. In few recalcitrant plant species, incubation of the source tissues or the donor plants in the dark may help in improving the protoplast yields as well as their culture efficiency or culture responses. All these treatments may or may not be required and would vary among different plants or also between the different tissues of the same plants too. Next is the culture types. That is, to use the semi-solid media for culturing the protoplast or the liquid media. So first is the agar culture, that is protoplast culturing on the semi-solid nutrient media. Agar is most frequently used for the solid support in the plant culture media. However, the concentration of the agar should be in the range that it forms the soft agar gels specifically for the protoplast culture. Higher concentration of the agar or the hard agar gels may impact the protoplast culture negatively. Usually, Bregman's plating technique is used for plating protoplast in the media. The protoplasts are suspended in the suitable liquid media at a density usually twice the desired one. Sterilized agar media is usually maintained at the required temperature somewhere around 35 degrees Celsius, and now both the media and the culture, they are mixed in the equal volume and then they are spread in the desired vessel, usually the Petri plates. The cells remains embedded in the soft agar medium, and therefore change in the concentration of the agar in the media has significant impact. Protoplasts remain at the fixed position, divide and form the clones of the same cell. Additional advantage of the culturing protoplast on the semi-solid media is that clumping of the protoplast may be avoided. However, some plant species do not respond to the culture in the semi-solid state. Further, type and concentration of the gelling agent can also influence the development of these cultures. In addition, the sensitivity of the protoplast to the temperature may also vary among the different plant species. In many plant species where the protoplasts are sensitive to the temperature, usually alginates are used as gelling agent. Next is the liquid culture. That means solidifying agents are not used and the protoplasts are cultured as suspension culture. The culturing of the protoplasts in the liquid medium has several advantages over their culture on the solidified media. First and foremost is that any manipulations would be easy to execute in the liquid cultures, like replacing the old media with the fresh media or diluting the inoculum for subcultures. Depending upon the type of experiment, protoplasts may be required at different densities. Hence, in the suspension cultures, it would be easy to make the dilution to adjust the density of the protoplast as per the requirement. For the maintenance of the protoplast, osmotic pressure on the culture media needs to be carefully maintained. In the beginning, we discussed about how the hypotonic solution can result in the bursting of the protoplast. Hence, it is easy to manipulate the osmoticum in the liquid cultures. Further, the protoplast may have differential responses on the agar culture versus liquid culture. It has been reported that in some species, protoplasts do not respond or grow on the solid agar culture at all, and therefore culturing the protoplast in the semi-solid state or the media is not even an option for these plant species. In some plants, protoplasts may release some metabolites in the culture media, and their accumulation in the media may be toxic to the growth of their own protoplasts. Therefore, change in the media may be required, and it would be easier to renew the media frequently if the culture are maintained in the suspension culture or the liquid cultures. Also, the nutrient requirements in the protoplast culture may change with the physiological status of the culture, and hence nutrient may be required to adjust frequently depending upon what development stage of the culture is. These adjustments can be executed easily in the liquid cultures. Liquid media also allow the rapid diffusion of the nutrient components in the media and hence keep all the protoplasts in a homogeneous state of nutrient availability. So overall, culturing protoplasts in the liquid media has several advantages as compared to their culture on the semi-solid state, and liquid culture are more popular. Next is the culture media composition. Now, nutritional and hormonal requirement of the cultured protoplast constantly vary depending upon at what step of the regeneration state culture is. Generally, the nutritional requirement for the culturing protoplasts are similar to the plant tissue culture, and usually MS salts with B5 vitamins are used for this purpose. However, there are few special requirements for the protoplast culture and their maintenance in the suspension cultures. Calcium is required at the higher concentrations. Usually 2 to 4 times higher concentrations are required as compared to the media used for the in vitro regeneration cultures. This is specifically required for the membrane stability of these protoplasts. Presence of ammonium ions may be toxic in some species. Therefore, either the ammonium nitrates or ammonium salts should not be used in the media, or if they are used, they should be used at very low concentrations. Also, the reduced concentrations of iron and zinc in the culture media have been shown to give the better results. Now, the exogenous supply of the growth regulators is always required almost all the times. The plasmolysing conditions usually suppress the photosynthetic capacity and even the respiration rate too. And hence the supply of the exogenous source of the growth regulator for the initiation of the cell division is mandatory. However, after the formation of the cell walls, exogenous supply of the growth regulators for their division may not be required. Usually, the growth hormones are required in high auxin to cytokinin ratios for the cell division. However, in cereals, only 2,4-D is sufficient for the induction of cell division, or even it performs better when it is used with little amount of cytokinin. Overall, the nature and the relative concentration of the growth hormone may vary among the different plants and need to be optimized experimentally as per requirements. If the in vitro regeneration system is available for a plant species, it can give some clues about the composition of the media required for protoplast culture. However, that is not always true. Like the callus culture in the cereals require higher concentrations of phytohormones. However, the protoplasts of the cereals are sensitive to higher concentration of the growth hormones, and hence optimization are required experimentally. Similarly, any carbohydrate source can be used for the in vitro cultures. However, the use of glucose is usually preferred and perform better than the others like the sucrose, fructose, maltose, and so on. And finally, the osmotic stabilizer. Presence of the osmotic stabilizer is essential for the maintenance of the protoplast. However, ionic osmotic stabilizers may interfere with the cell wall developments. Overall, the nutrient composition has a huge impact on the growth and culturing of the protoplast and should be optimized carefully. Next is the culture medium osmoticum. Now, in the natural plant cells, protoplasts are always protected by the presence of cell wall. However, in the absence of the cell wall, protoplasts are naive and need osmotic protection in the liquid medium. Maintenance of the right osmoticum is therefore crucial for the maintenance and culture of the protoplast. Generally, osmotic stabilizer used in the culture media can be grouped into two categories. One is the ionic components and another one is the non-ionic components. So the first one is the ionic components. Inorganic salts are supplied in the form of various salts like potassium chloride or calcium chloride or magnesium sulfate. These salts ionize in the liquid media and are ionic components that regulate the osmoticum in the medium. Hence, any change in the concentration of these salts will alter the osmoticum of the media too. Carbohydrate sugars are the non-ionic substances that influences the osmoticum of the culture media. Carbohydrate sugars are also two types. One are the metabolically active sugars and another one is the metabolically inactive sugars. Metabolically active sugars are those that are continuously used by the growing cells as an organic carbon source and are metabolized. On the other hand, metabolically inactive carbohydrates are those that are not used by cells as carbon source, as cells are unable to metabolize these sugars. Sucrose, fructose or the glucose are the metabolically active sugars, whereas the mannitol or the sorbitols are metabolically inactive sugars. In an actively growing culture, the metabolically active sugars or the inorganic nutrients are continuously used by the growing cells, and therefore the concentration of these components decreases with time, and this may result in the change in the medium osmoticum. Therefore, use of the metabolically inactive sugars is advantageous and crucial, as these are not utilized by the growing cells and therefore these work as an osmotic stabilizer in the culture medium. Next is the application of protoplast culture. Since 1960s, when the enzymatic methods of protoplast isolation were optimized, protoplasts have been used for large number of applications to study the basic biological phenomena or the commercial application in the product development. So first is the study of basic biological phenomena. Now the protoplasts are the good system to study the impact of environmental factors on the osmotic behavior of the plant protoplasts. Protoplasts burst open quickly if placed in the hypotonic solution, or even if these are dropped on the glass slide from a little height, these break easily. And therefore the plasma membrane preparations can be made easily and can be used for various investigations to study the plasma membrane structure and function. In the absence of the cell walls, exposed plasma lemma can be studied in great details for various membrane associated functions like transport, uptake mechanism, permeability, membrane fusion mechanism, or the relationship of the plasma membrane with the cell wall. Protoplasts can be used to study the cell wall synthesis, the deposition of the cellulosic microfibrils and their orientation on the protoplast surface, how these are initiated or how these are placed in the beginning and how the polymerization proceeds. These all phenomena can be studied using the microscopic analysis of the samples collected from different time points during the cell wall regeneration. Now, protoplasts are an excellent resource for the extraction of various cell organelles like nuclei, chloroplasts, mitochondria, and so on. Similarly, the protoplast has the ability to take up the isolated organelle through the process of endocytosis. Therefore, uptake of the organelle like the chloroplast offers an excellent system to study the interrelationships or the interactions of nuclei, chloroplasts, and cytoplasm brought together from different plant species. Protoplasts are a good source for studying the various biochemical or biophysical aspects of the photosynthesis or various morphogenetic responses in plants. Overall, an isolated protoplast provide an ideal single cell system for studying the basic biological phenomena in plant sciences. Plant cells are totipotent and hence the protoplast extracted from these cells are also totipotent and they can be regenerated into the whole plants. However, their capacity to regenerate would depend upon several factors like the physiological status of the source tissue, health or age of the plant, or the mitotic or metabolic status of these tissues. Because of the capability of totipotency, genetic manipulations can be performed as these are easy to operate in the isolated protoplast as compared to the mature cells. Delivery of the foreign molecules is usually prohibited by the presence of cell walls. However, because the protoplasts do not have the cell walls, therefore these manipulations like the delivery of DNA, proteins or other biomolecules in the protoplast is much easier, and after the required manipulation, whole plants can be regenerated. Somatic hybridization is another important application of the protoplast where we can fuse the protoplast from two different plant species and regenerate the hybrid plants. We will talk about the somatic hybridization in a little more detail in our next lecture. Protoplasts are a good system to study the interaction between the plant cells and viruses. In general, the infection process in plants is not synchronous and the infection establishment is also difficult. Usually it is restricted to the cells with damaged cell walls. However, protoplasts can directly inoculate it with the pathogenic viruses and infection can be more or less synchronous. It can help to study the process of uptake of the virus particles by the protoplast, virus replication in the protoplast, or their mode of action at the molecular level. Further, induction of somatic embryogenesis have also been achieved from the protoplast, and the direct somatic embryogenesis is used in commercial micropropagation and development of the artificial seeds. So in this way, Protoplast has played a significant role in the basic biological applications to study the basic biological phenomena, and also they are used in several commercial purposes. So in summary, we talked about the protoplast and various methods of protoplast isolations. We also talked about the various culturing requirements for the protoplast cultures with respect to the type of culture like liquid culture versus the semi-solid cultures, biological parameters of the source tissues, culture media composition, or the osmoticum in the liquid media. Finally, we talked about the various applications of the protoplasts in the basic biology as well as their commercial aspects. In the next few lectures, I will discuss more about the commercial applications of the protoplast. Thank you.